As part of this specific immune system, one group of cells are an important part of how the immune system responds to pathogens. These are the B lymphocytes. By now, you should understand that the majority of B lymphocytes will be found in the lymphatic system, particularly in lymph nodes, which are found in various places around your body, such as under the armpits, groin area. What takes place here is that a result of something you should have studied already, where phagocytes have engulfed and digested pathogens, and some of them may well then go on to become what we call antigen presenting cells. So this will be a phagocyte, perhaps a macrophage, and on its cell surface membrane will be an antigen, here indicated like a red triangle, such as what we have found on the pathogen itself, in this case this bacteria. The antigen presenting cell will migrate through tissues, blood and lymphatic system, and will make contact or come across various lymphocytes, whether they're B lymphocytes or T lymphocytes. Inside the membranes, or in the membranes of lymphocytes, there will be antibodies acting as receptor, which I'm trying to represent with these objects here. I'm showing them with different shapes because each different B lymphocyte potentially will respond to a different shaped antigen, depending whether the antigen fits with the antibody in the membrane. If you remember the key word there, it's complementary fit. So in this case, this antigen presenting cell does not complementary fit with the antibody in this B cell lymphocyte, but it does with this one. Now each B cell, each B lymphocyte is a clone. This is a different clone from this one. And it's because this one has been selected, we call that clonal selection. That one B lymphocyte has been clonally selected, only because this antigen presenting cell has no joined to the antibody with the antigen. What takes place now is that this B lymphocyte is now triggered to divide. Because this one B lymphocyte was clonally selected and is now dividing, this process is called clonal expansion. Other key words to describe this process is that the B lymphocyte is rapidly dividing by mitosis. So we get a large number of these B lymphocytes, hence clonal expansion. So here we have the large population of that particular B lymphocyte. Some of these B lymphocytes will now remain in your body in quite a large number, perhaps for the rest of your life, certainly for several years. And if you are exposed to the disease again, your B lymphocytes will respond much quicker than the first time, which is why you build up some immunity. It's almost as if your body remembers the exposure to the pathogen or vaccination, which is why these type of B lymphocytes are now called B memory cells. The way to describe it is that you now have immunological memory. Other B lymphocytes will specialize. They will differentiate into what we call B plasma cells. And I'll explain the role of B plasma cells on the next slide. B plasma cells form something which is called humoral immunity. It's quite an old term, but you might find it in some textbooks. But what they do is that they release antibodies. These are all antibodies that are specific to that particular antigen that was presented to the original clone. So any pathogens with that antigen on, these antibodies are going to complementary fit to it and stick to it. The structure of antibodies and how antibodies work we'll discuss another time. One statistic I've heard is that plasma cells, plasma lymphocytes, potentially release up to 2,000 antibodies a second. They are proteins, which is why it's vital that you have correct nourishment, especially protein, to have an effective immune system. So people who are malnourished or hungry, going starving, they tend to suffer more diseases. Make sure you watch the next video, if you haven't done so already, of clonal selection of T lymphocytes.